Okay. Good morning to one and all present here. I, Dr. Vishal B. Jadhav, Associate Professor, Department of Pharmacology, welcomes you all for 1 week online international e-FDP, e-STP on empowering pharmacy education, navigating regulations, global trends, and advancing outcome-based education and research organized by Internal Quality Assurance Cell of Gokhale Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, in association with Association of Pharmacy Teachers of India. Today, we have with us an eminent personality, Dr. Ozair Alam, Professor, School of Pharmaceutical Education and Research, Jamia Hamdard University, New Delhi, as a speaker of session 1 of day 5. I heartily welcome you, sir, and thank you for accepting our invitation to address the gathering. I am here to introduce you, Dr. Ozair Alam, sir. As far as educational qualification concerned, Sir completed his PhD in pharmaceutical chemistry in 2010, and his research topic was design and synthesis of novel six-member heterocyclic compounds of pharmaceutical interest. He done his Master of Pharmacy in Pharmaceutical Chemistry in 2004 with a gold medal, and his Bachelor of Pharmacy was completed in 2002. Sir has more than 16 years of teaching experience at B.Pharm and M.Pharm level. Currently sir working as professor in Department of Pharmaceutical Chemistry at Jamia Hamdard University, New Delhi, India. He was worked previously as associate professor in Department of Pharmaceutical Chemistry, Jamia Hamdard University for 4 years, and assistant professor in the same department at Jamia Hamdard University for 12 years.
No, 9 years.
Okay.
9 years only.
Yes, sir, yes.
I've been professor from 2021.
Yes, yes. As far as his award and achievement concerned, Sir name was included in the list of top 2% scientists by Stanford University, USA. Sir received prestigious DST Sub Young Scientist Award in 2013, received gold medal in 7 convocation for securing first position in MPharm in 2005, received first prize with merit certificate for securing first position in MPharm. Sir was selected for JRF by UGC during the period of his PhD. Sir received Best prestigious Dr. P.D. Sethi Annual Award in 2010 for Best Research Paper on application of PLC, HPTLC in pharmaceutical, herbal, and miscellaneous analysis. Sir qualified GATE in 2002, conducted by Indian Institute of Science, Bangalore. In concern with the publication and presentation count, Sir successfully guided more than 41 PG students, successfully completed more than 4 research projects. His publications in renowned international and national journals. Sir published more than 116 papers in national as well as international journals and published more than 4 patents. His Google citation scores is more than 3,000. His citation is about 3,690. H index is 33 and I-10 index is 71. So I welcome on the behalf of IQAC cell of MAG Copper and APTI, on the behalf of these cells, I welcome Dr. Ozair Alam sir to deliver his expert talk on the topic in silico approaches in drug discovery and development. Thank you very much, sir, again for accepting our invitation.
Thank you, doctors, for giving wonderful introductions. Very, very warm welcome and very good morning to all. So today I'm going to talk about a very important topic that is drug discovery and development. I'm audible to you all?
Yes, sir. Yes, sir. You are audible.
Yes, yes. Okay. So I'm just going to talk about a very important topic, that is in silico approaches in drug discovery and drug research. So we lot heard about docking, pharmacophore modeling, QSAR, And nowadays AI tools and machine learnings and all, because these things might be outdated within 5 to 6 year or 10 years, because you just have to put the large databases of structures in the AI, and that AI giving you the all, I mean, outputs of what you desired from that. So first we come to the why we have to dock first of all. All of you know that the drug which has, why we can say the any compound which has the binds with some receptors, enzymes, or something like that, or target site, because the drug has the affinity for binding it. So in protein ligand docking, basically the software we have used for docking of these compounds is composed of two parts, which that can work together. So first one is called as the cell search algorithm. So algorithm is a kind of mathematical equation or something like that, which can give you the, I mean, some certain values and a lot. So suppose here I am taking one example son of I'm ongoing of my one of the students SRF research project. She has taken the chicory acid as a ligand. So this is the structures of ligand. Already we know that this chicory acid having rotatable bond. So this is the phenyl side on that side. So whenever we put, I mean, prepare a ligand of this chicory acid by lake preparation. So what we saw that we have to generate large number of conformers of chicory acid, and these conformers is actually having a different shape and size. Why they have to form a different conformers? Because the rotations of these, by changing the dihedral angle, by changing the torsional angle. So due to these changes in the chicory acid ligand, it can form a different types of conformers, and these conformers have the different shape, and each conformer has ability to bind with the target site in a different pose or different these conformers of chicory acid bind with that of the one of the site that is called as the alpha amylase. This is the sites of the, I mean, for target for development of anti-diabetic agent like alpha glucosidase or alpha amylase. So this is the binding cavity here, where these two most potent conformer is just going to bind, and just we put to bind in that. So this is called as the search algorithm. Next, the next part of this ligand protein docking is after search algorithm is the scoring functions. So we have to score, we have to rank that particular poses or conformation of that ligand, which is from the rest. So already we know that the drug or any molecule which has the binding affinity, due to this binding affinity, it can binds with the protein or the target site. So basically here what we do, because if suppose I am coming to the department, I say hello and shake my hands to some of my colleagues, and they just giving my warm welcome. Or so whenever I shake the hand of that particular colleague, so what happened? There's an exchange of these energy. Sometimes we just tightly hold this the hands with my colleagues, or sometimes with the loose grips. So that's why there's exchanges of this energy. So here what we do, we just have to identify the binding modes of that particular compound. So basically when the ligand bind with that of the receptors or binding site, they have to calculate certain interaction energy between the ligands and the protein receptor. And these interaction energies, some of the van der Waal, some of that van der Waal interaction, electrostatic interaction, hydrophobic interaction, and hydrogen bonding interaction. So some of these are non-covalent bond, all that. So according to these energy, those who have very less energy, they have more stable in the more stable conformer, and they are more stable in the binding site. So this is called as the scoring function. So when we talk about the glide docking, glide is that called as the generalized ligand docking. So here, what we do, we have the three type of ligand docking. One is called as the high throughput virtual screening. Suppose you have the millions of that compound which you have take from the any database, and you have planned to do a dock of all these and screen out only few compounds from these databases, like Jing database, drug database. There are a lot of databases is there. So in that case, we just run these all millions of ligand in the docking panel. So the each compound, yeah, each ligand took only 1 to 2 seconds to complete their docking studies. So, but once we have to do standard precision, where the 5 to 20 second is required to complete the one ligand dockings output. Then we come to the extra precision, where each ligands will take 2 to 3 minutes. So means here extra precision means that means more accuracy about that particular ligands and their conformers. So once we talk about the high throughput virtual screening, although it has the high speed and it take less time to complete the dock, but here the accuracy or correctness is less. But once we talk about the standard precision, but during the virtual screening of large library, we should have to start always from the high throughput virtual screening, because you cannot do the dock by taking millions of compounds by extra precision. So that's why we have to screen out from 1 lakh or 10% of it, then by SP, then 10% of it by 1% of that. So finally we got 5 or 10 hit from this large library. So so basically, in molecular docking, we just have to identify the molecules on the basis of their binding ability to their target or the receptor. So there are two types of basically docking. One is called as the structure-based drug design, and that is called as the ligand-based drug design. The structure-based drug design means what? If I lost my key and ask the key maker to please prepare the key. So what the key maker, he just see the structures, dimensions, and that part of cavities of that particular lock before going to prepare the key of that. That is called as the structure based, because where we have to know the target structure, 3D structures of the design. Like suppose if I am talking about the EGFR kinase, so we must have to know that where it is the binding pocket, hydrophobic, hydrophilic, where it is the hydrogen bond area, hydrogen bond domain, and all that, etc. So we have already know the 3D structures of the receptor or enzymes or any protein. So according to that, we have to fit the ligand into it and dock it. Okay, so basically in case of structure based drain, that is the molecular docking. One is called as the molecular dynamic simulation. Then we come to the ligand based drug design. So in ligand based drug design, so either we have to search the 2D QSAR, where we have to generate the mathematical equation between physicochemical parameters and biological activity, because biological activity is expressed in log 1 upon C. C is the minimum concentration required to define the maximum biological activity in 2D QSAR, like Hans equations, like Hammett, like all Taft steric parameter, all this we have to develop in that. And once is called as the shape-based 3D QSR and the pharmacophore analysis. So we know that particular functional group in that particular molecules, like some of these are aryl, the hydrogen bond donor, hydrogen bond acceptor, some of the cationic, anionic. According to that, we have to develop. So here in we know the structures of the ligand without knowing the target site, which is the independent of any information about the molecular target or receptors or enzyme. So already we know that docking is basically computational predictions of binding mode and binding affinity. One is that binding mode means how the drug or any molecule binds in what pose. And binding, suppose if I'm sitting on this chair, so which is more comfortable that positions of my chair. So that is called as the binding mode and binding affinity between the protein and ligand. So basically, docking helps us to find out the probability of interaction between the two biological molecules or between one biological and another small molecule. So sometimes we talk about the protein docking. So these are two biological molecule, and one biological molecule, either the receptor, protein, enzyme, or any target site, and another is your ligand. So that's why we have to establish an interaction between the ligands and the target site, and then is the interaction maybe with the two protein or maybe with the one protein and one ligand molecule. So docking actually provided the binding pose. Pose means what? Conformation and the orientations of a ligand in the binding site of biological macromolecule by maximizing interaction while minimizing the total energy of the complex. Because once we have to minimize the energy of the ligand, so we have to correct the bond order, bond length, and all by OPLS method. Because by OPLS forceful method, because how the drug is behaved, like suppose we talk about the carbon carbon single bond, what is the distance of that? 1.44. Suppose we talk about the carbon carbon double bond, we talk 1.2 something is that. So already this set algorithm is there for that. So this by the OPLS forceful methods. So here you can see that this is the protein, the whole protein we have taken, and we just try to bind this ligand on the active binding site of that. So they have to form a protein ligand complex. Uh, Now, if whatever we perform the docking, which is basically the rigid docking. In rigid docking, what we do, your receptor is basically have the 3D structure which are rigid, which are not flexible, but your ligand is flexible, which can bind according to that the structures of that particular rigid. So, if you perform rigid docking of any of your design molecule, but you cannot got I mean sufficient output data or sufficient energy or sufficient docking score. In that case, you have to perform the induced fit docking. So here, induced fit docking means what? Suppose I have taken one examples of here that catecholamines or something like, which is just trying to bind with that receptor, and these receptor having van der Waals interactions here, and they make a hydrogen bonding, and they make a here ionic interaction with the aspartic acid or something. So you see the distance between these CO minus and I mean quaternary ammonium group is large. So in that case, you can say very weak or interaction. So what does do when the ligands bind in the binding site, the receptor itself change in their shape to so that the carboxylate ion get closer enough to that of the quaternary ammonium. So they have to make a strong ionic interaction. They have to make strong ions. So that is called as the induced fit dosing. So that's the we have to change the site of geometry of protein complex depends upon the conformational changes induced by the bound ligand. So here the ligand has property to change the size and shape of that receptor. So induced fit protocols solve this problem by existingly consider possible binding modes and the associated conformational changes within the receptor actives. So each pose structure prediction is then used to accommodate the ligand by reorienting nearby the side chain. So these residues and ligands are then minimized. So what we do, suppose this is the side chain of amino acids of any of the binding sites. So we have to reorientate that so that these make a stable interaction with that of the ligand. So that's why induced fit docking is always been performed when we did not get a desired result by rigid docking. So in that case, we put the ligand through the induced fit docking, and the receptor itself change their shape and size. And after that, we come to the covalent ligand docking. So some of penicillin derivatives like beta lactam antibiotic like ampicillin, amoxicillin, cloxacillin, dicloxacillin, because these antibiotic is have a very big problems like because their lactam ring is basically open up by acid attack or by the or by the beta lactamases. So that's why the scientists who were put some electron withdrawing group here, NH3 plus, so which withdraw the electron density from the nucleophilic oxygen, so they less likely to attack on the electrophilic carbon. So that's why the bond is not lactam ring is not going to open. By this way, we have to develop a stable beta lactam antibiotics against the acid attack. Similarly, if you put some of the bulky group, so that's why beta lactamases, the penicillinases, but not get close enough to that of the lactam to open up the string. So basically, if you talk about that penicillin beta lactam antibiotic, which has the binding to the transpeptidase enzymes of penicillin binding protein, so they have to acylate here, and they make some covalent interaction, and this covalent interaction will not detach till the new receptor or new enzymes is synthesized. So that's why this is the way how the cell wall synthesis is just going to stop by the drug binding with the covalently with that the target penicillin binding protein of transportase. So they have to inhibit the cross-linking with that of the NAM and NAGs. Already we know that. Similarly here, they also, because the penicillinase is also going to degrade by the beta lactamase or the penicillin, the same way they have to acylate on that. Similarly, the covalent docking, most of that EGFR kinase or CDK inhibitors like afatinib, apitinib, osimertinib, so they have here the certain double bond is here. They have to make a covalent interactions with that. For covalent interactions, some of the residues like serine, serine or like cysteine, so they have to make a covalent interaction with that of the molecule. So that is we do with the covalent docking. Covalent docking is giving a, most strong, because here the drug will not ever detach from that receptor. So rigid docking we discussed, induced fit docking, covalent ligand docking is there. So now one of the tool which where we have used to identify the binding sites in that of receptor. Sometime you have to open up the PDB from the protein data site. So what we see, we never find any co-crystal structures. Never find any co-crystal structures. So how would I know that where we are just going to bind our own design molecule in that particular protein? So by this we have to use some sitemap analysis. If you are not going to perform the sitemap analysis, there is one tool that is called as the homology modeling. So where the binding site in homology, the protein which we have taken, they don't have any binding site, and we just have to complementary with binding with by the last site. So those we have taken some of the species which have the co-crystal ligand, and we have to match the sequence of that amino acid to the homology modeling, and then we have to make a model, and then we just try to bind our molecule through the dock. So in site map analysis, we have to identify the binding site on the protein. If you don't have any information about the ligand in the protein structures, if you don't have the information about the binding pocket residue, so you can identify the top rank potential receptor binding site. If you don't have any co-crystal ligand present in the binding site, then you can perform the sitemap analysis. And sitemap analysis is also help us to understand the binding site properties, which can be used for the lead optimizations. Okay, so use 3D structures of the target receptors to design such potential drug compound that can be modulated. So here I'm taking one receptor that is called as the EGFR kinase. So in case of EGFR kinase, you see there's the whole protein structures of the EGFR. So they have 5 binding site. Here they have the 5 binding site, that is 1 2 3 4 5. This one is that is the first of all, what we have to do, we have to prepare a protein through the protein preparation wizard, and then after that we have to run for sitemap analysis, and through the sitemap analysis we have to get a 5 grid here, 1 2 3 five receptor grid. And after that generation of grid or site map, we are not going to generate the grid of all these five sited. We have to generate a grid of the top score. So in sitemap 1 we have the site score 1.0, this is above 1, and D score means drug ability, and V is the volume and is the size. Drug ability, yeah, drug likeness character of that target site. So out of these five sites which have the high D score or site score, obviously the site map 1. So we can say this is the actual binding site of the ligand of the EGFR kinase receptor. We are going to discard all these site map 2, 3, 4, 5. So we have to prepare only the receptor gate of these sites. Okay. So here already we know that here we just after preparing the receptor grid, we dock with this ligand at that particular site. So we can see these are the 3 or 4 area. Yellow area is called as the hydrophobic area of the receptor, and here the blue area, that is called as the ligand hydrogen bond donor region, and red area is called as the ligand hydrogen bond acceptor region is there. So you can see methionine 769 cysteine aspartic acid, and here is that uh, red color which denoted the ligand hydrogen bond acceptor region, which is binds with that of the license. This paper has been published in Archive the Pharmacy of one of my PhD scholar in 2018. So what actually, how to calculate the D score or site score volume? So site, am I audible to you? Yes. Hello. Can you hear me? Hello. Hello. Can you hear me? Hello. Can you hear me? Hello. Hello. No one is there?
Sir, you are audible, sir.
Yeah. Can you hear me? I mean,
Yes, sir. Yes, sir.
I just asked you for some any response. Nobody.
Yes, yes.
So, so site score is basically here we have by this applying this formula, dragability score by D score. With that, here N is called as the number of site point, which is denoted by some of the white sphere, and N is the number of site point, which is denoted by, and we have to cap if your site point is 100. Then E is the enclosure score and P is the hydrophilic score. By this, we have to calculate the D score, site score. If we got the top rank, then we have to choose for docking. Okay. So D score, dragability, which is more than 1, considered as a draggable site or binding pocket. If value is less than 0.70, then we have not considered as a binding site. If site score is more than 1, considered as a dragability site or the binding pocket. So during the COVID time, during the COVID pandemic of the coronavirus 2019, which is caused by the severe acute respiratory syndrome, which is the global crisis, and we know here we have taken one of the site that is called as the RNA-dependent RNA polymerase to develop. So through the site map, we have to identify the 5 site. Site 1, site 2, site 3, and 4, and 5. And after that, we dock with this site 1. We probably see the binding of these molecules. Next, we have to perform by the sitemap analysis is that uh, we have taken one compound that is called as the swartirine, which is obtained from swartirine plant, and they have a very interesting activity against. They shows some cardioprotective effect. They shows from anti-cancer effect, and they shows some anti-inflammatory or antioxidant effect. So suppose we have taken the two enzyme that is called as the superoxide dismutase or catalase. These two enzymes is responsible for, because in case of uh, uh, in the body, if the free radical is generated, so which form a oxygen dianion as a free radical. So these free radical may have to modulate our body cellular functions, so they can cause cancer, they can cause inflammation, and they can cause some restrictions in the cardiac cell. So how to avoid and how to swartirine is act as an antioxidant. So super disoxide dismutase, we just convert these, what's called as that uh, O2 anion free radical by super disoxide, which is a zinc copper enzyme. So they just convert into that of the hydrogen peroxide and oxygen to less toxic or some things. Is that after, in the next step, we have to form by the reaction by catalysis. So catalysis converted the hydrogen peroxide into the oxygen and water. So by this way, we just have to eliminate this free radical from our body by antioxidant effect of the swartirine. So on the right panel, we can also perform the sitemap analysis of the superoxide dismutase. Here, they just have to see zinc and copper, which is involved in for catalysis purpose. This is the dimer protein. Here, this the drug is actually swartirine is bind in between these. And on the left panel, you can see here is that tetramer, uh, pentamer protein is there, catalysis. So this is the binding uh, pocket of that. So now, this is the very important things we have to discuss now. If somebody who is working in the area of pharmaceutics and they just want to see the synergistic effect of these two drug, most of this they have taken one synthetic, one uh, naturals. So here we have taken one drug is celemerine, another is the epigallocatechin gallate, okay, which is obtained from the tea and celemerine. So how do we have to get the synergistic effect of these two drug in the nano formulation? So for that, how by docking we have to establish a hypothesis of synergism. One, we have to take in the ATP binding site, another is called as the allosteric binding site. So basically, here we have taken the EGFR enzyme target for development of anti-cancer potential of these two drugs in synergism manner. So this area is called as the allosteric binding site. This is the EGC. Why we have chosen the allosteric? Because most of the drug which is in the ATP binding site, they can cause some like resistance. So if you choose the allosteric, the resistance is just going to be overcome. So here we have chain A, B, C, D. So here we have to perform the molecular docking at the ATP binding site as well as the allosteric binding site. What we see, these two drug binds at the, here this is the ATP binding site, here this is the, so all epigallocatechin gallate and celemerine were bind with the different binding site amino residues of like methionine 790, which is the hinge region of that EGFR 790, and here aspartic acid and all that. This is the area of the binding sites of receptor surface of the celemerine, and this is the epigallocatechin's binding sites. And once we have to perform the dock of these of synergistic effect, then we have to superimpose, because these ATP binding site which have the co-crystals binding. So we have to superimpose superimposed co-crystal with that of the silyl. You can see the binding orientation and pose is exactly same as that of. Okay, so in that case, we got a very good result is there. You can see the our silymerine is perfectly, I mean, orientate and perfectly have the same binding orientation and pose with that of the co-crystal ligand and with the same binding residue. And silymerine have the Dock score is -11. Okay, uh, sorry, co-crystal, and our silymerine has the Dock score of that. Then similarly, we have to perform the superimpose structures of that our co-crystal ligand at the allosteric binding site with ECGAP gallocatechin gallate, and we can see the binding orientation is same as here, the methionine 793 and the aspartic acid and all that. They are same, and the Dock score is very high. Smartini is it and so now this work is, I mean, we just perform with that of one of his scholars of that Professor Javed Ali. So it's under communications, this part. Now we come to the, I have just published two paper in around, I think, 2020 or something, the 21, where one of my scholars, DST inspired, she is currently in UK. So she has performed and she has designed our molecule by the de novo design. So if you see this part uh, of that, they have the different parts is there, green, blue, I mean, pink and all that. So this is our design molecule, and these all are the co-crystals or potent molecule taken from the literatures, and we have to generate one, I mean, hybrid molecule by by different component, and that by finally we have to synthesize this compound, and we can restore the interaction by hinge region methionine 793. This is very crucial for the activity. You can see all these potent compound or co-crystal, they have same interaction as by design compound. So you can see this is the hinge region. So de novo design is the design of bioactive compounds by constructions of ligand model, then model of receptors, enzyme active site, structures of which is known from the X-ray or NMR data. So this is this type of compound is act as a type 1 kinase inhibitor. By docking, how we have to say that your molecule is the type 1 inhibitor, type 2 inhibitor, type 3, type 4, or covalent interaction, or ATP binding or allosterotic binding inside. So here, this is the whole proteins of the EGFR. They have the different reason, shows that alpha helix, phosphate loop, activation loop, catalytic loop. This is called as the C loop. This is the N terminal, and N terminal join to the C terminal through a hinge region. This is the turquoise that is called as the hinge region. This is called as the ATP binding region, and this area is called as the DFG motif. DFG motif either in or out. D for aspartic acid and F for phenylalanine, G for glycine. These are the three amino acids which are in the sequence. So on the basis of this DGF motif, we can have to say our compound is type 1 or type 2. So we can see the right panel here on that. So in DGF in conformation, we have the side chain of aspartic acid. Okay, DGF motif points to the hinge region. So this is the hinge region, and here we have our own design compound and the co-crystal ligand, that is the gefitinib, and the another amino acid that is called as the phenylalanine. So side chain of phenylalanine is, which are away from the hinge region. So it means that our DGF motif is in conformer. In out conformer, what happened? The phenylalanine is towards the hinge region and aspartic acid is away from the hinge region. That we have to develop the type 2 in case of kinase 2 inhibitors or so in that case DGF motif out is there. So by docking, we can also say that our design molecule is a type 1 kinase, EGFR kinase inhibitor, type 2 or type 3 or covalent. So by docking here, we most potent compound, we can see they have the strong hydrogen bond interaction at 2.19, 3.216 as particles. So we can say there is a weak one interaction hydrogen, but is strong because through the glide, we just have the default mode of selecting the amino acid distance between the ligand and target is 2.5. In mass stereo is the 2.8, and in glide only the 2. So we can have to increase the distance of interaction between the uh, atoms of the amino acids of the binding sites and the atoms of the ligand, which are going to interact with that. So we can have to increase the distance to the bond. And one more thing is that these compounds superimpose with that zephyrine, and you can see this is the hinge region, and this is the hydrophobic pocket, and this is the front pocket. So we can see these compounds is aligned properly, and they can have to bind with the hydrophobic as well as the front pocket. Front pocket is also the hydrophobic area is there, and where we have to perform the molecular surface view of compound with that of the zephyrine, which have. And the next thing is that how to represent our docking data in research paper. That is very important. Is that so? Our aim is here. What you have to perform the docking of your design molecule till the desired result is not achieved. Desired result means what? Because every PDB suppose you have select 50 PDBI or 10 PDBI, and we have to generate a grid through the protein preparation wizard, and you have to perform the dock of all these. Okay, with taking starting from high resolutions to low resolutions X-ray crystals of that crystal. And once we have to perform the docking, what you have to observe or visualize, you have to visualize, because this is the hypothesis. So hypothesis must be strong enough. Hypothesis must be strong enough to establish that whatever I design, it may be the most potent drug candidate in futures, or after we can validate by in vitro and in vivo studies. So you can see how these our design molecule is have exactly same. So might be I have performed the dock of more than 50 20 or 50 PD, then we got this result. So we just not restrict to perform the dock by taking only one PDBID. If you get the good result, and we have to compare the dock score of co-crystal ligand with that of the your design compound. Your design compound must have high or maybe near to that of the co-crystal ligand. So here we just have to take in the different substitution and all that. Doc score is like that. So in that case, methionine and all that. So here in the table, what we have to present your data, you have to put the atoms, ligand which are going to interact, what are the amino acid which are going to interact, what are the distance of that bond, whether it is a hydrogen bond interaction, pi-pi interaction, pi-cation interaction, or maybe the hydrophobic interaction. This is the way we have to represent our output of the doc in the tabulated manner in the research paper. uh, this is the paper which we have published last year in archive the pharmacy. So we have already designed this molecule and we synthesize all. So it you have to see that our design molecule is this is the MAP kinase inhibitor, which is just used for development of anti-inflammatory drugs. So all these kinase, they have ability to bind with the hinge region which connect from the C terminal and N terminal. So they have to binds with that methionine 109. Similarly, in case of EGFR kinase, methionine 769. So they have to bind with the hydrophobic region 2. So you can see all these co-crystal structures of SB of 2A PDBID. This is the PDBID of 1ZL co-crystal structure CP844, and this is the and this one we have taken the PDBID of 1YWR. So in all four co-crystal structures PDBI, the orientation and binding pose is same as our design molecule or synthesized molecule. So this is the approach we have to apply in our research papers to develop a strong hypothesis. So now, recently we have to perform 2 months back, we have to perform resveritols and the lamotrigine. So that is the dual pocket inhibition. We are just going to develop a anticonvulsant epileptic potential of these two compounds. Lamotrigine, already we know that they are tracked by the sodium channel, and we have to take the resveratrol, is one of the famous and known, natural product which have a very large number of uses, either in cancer, in diabetes, in neurodegenerative disorders like Alzheimer's, Parkinson. So how? So here, these resveratrol is C site and big site lamotrigine by the sodium channel receptor. So what we do, we have to perform that how this resveratrol is actually binds with the site pore cavities of the domain site C. So once it binds it, there is no any influx of the sodium ion through this channel or through this pore, and there is no any generation of action potential, no any generation of your neurons or nerve in the CNS is not going firing high. By this way, you can say they can show some anti-epileptic, yeah, anti-seizure effect. So that's why this is the dual pocket inhibitions of resveratrol and lamotrigine, which maybe come within few months in the publications. This is under communications. So I have taken only the recent things, not very past what we did. This is, very good those who are working in the area of pharmaceutics, because in the pharmaceutics, the workers, yeah, researchers who has always taken two drugs in synergy, and they have to make a nano formulation. So before going to perform your in vitro in vivo study, you must have to develop a strong hypothesis of dual inhibitions or dual pocket inhibitions in case of epilepsy. So here, this is the dog pose of lamotrigine and resveratrol, the same binding sites. So they have. And we also perform this resveratrol, because resveratrol has also ability to bind with that of the GABA. This is under also the communications. GABA binding site, GABA A receptor, they have to modulate and influx of the chloride ion due to hyperpolarization. Here, once the ligand is bind, the pore is open, and they cause the hyperpolarization. So by this way, shows the anti-convulsant, anti-epileptic effect, because in absence of your compound, the pore is closed. Okay, the pore is open, pore is. So this is the electron cryomark of the different chain. Is that this is because basically GABA is the pentameric chain is there, 2 beta 1 alpha 2. So here the ligand binding site is available in between that. So in docking, we have a very important docking that is called as the virtual screening. We have to perform because this paper has published in 2023 in Journal of Biomolecular Structures, Dynamics. So here we have taken a billions of compound from the library, delivered in 1 week. So here we have performed 3 model by taking 21. This is for the chemical data emissions. We have synthesized the 50 compounds from the lab. Okay, and test set, training set, and then we have to generate a 3D hypothesis pharmacore. So based on that score, we have to take this model scene, and that we have to perform the ligand-based drug design. And so we know the structures of the pharmacophore of that particular structure, and we have to design 5,000 molecule from that. And we have to prepare the ligand, and after preparation, we have to generate the grid. We have to perform the virtual docking, same as I explained. We have taken 5,000 by height, then standard, then extra precision we have taken. And from after dock extra precision, we have to generate only the 5 hits from that. So we have to combine all these synthesized compound with that of the model generated from the libraries there. And after mmGVC energy. We just perform the molecular dynamics and we get the further perform the in vitro alpha glucosidase as well as in vivo anti-diabetic effect on animals. So this is the different harmful, and this is the how we can say that which state is active, which state is inactive. So they have the different pharmacophore hypothesis. One have the aryl, two aryl ring is there. Here is that R, three aryl ring is there. Here is the acceptor, root donor is there. So this is the static aspect of, because in the field based, or we have to see the static electrostatic interactions. So this is the red area. So blue area can enhance the activity and red area can decrease. So we are not going to substitute anything on this area. We can substitute any part or any moiety in this area because this is the favorable, and this is the yellow part. So electrostatic features and hydrophobic features is here in that assigned as a yellow area is there. So once we have to get a model, in future we have to design different libraries for future predictions, and we can validate by in vitro and in vivo studies. So somebody who is working in the area of natural chemistry, what they have to do, suppose one of my SRF student, they are just working on the chicory acid, or they perform alpha glucosidase. So they have to find out the ligands from that plants, and we perform the dock of these all compounds. So from this luteolin and epigenin is found to be more active, and we can see that dock score and all. We can see the different interactions. So this is also the area of DNA topo resveratrol. This is also the under communications, doxorubicin, and so here you can see that how can you say that DNA topoisomerase is actually responsible for elongations of the chain. So if we are going to stop inhibit this topoisomerase second, so here the chain is not going to elongate. So the cleavage of DNA, so which we have to perform by the docking. So this is the these are the, I mean, you can say these different types of nitrogenous base, adenine, guanine, thymine, cytosine. They have to make a hydrogen bonding with that of the so they have the sequence. So here this area, there's the chain is just going to be cleavage by stopping the DNA topoisomerase second, which is this this is under communications. So this paper has been published in 2017. I think 3 paper which we have published in PPR gamma modulation. So 2 is in bioorganic, 1 is archived pharmacology. So one of my project DST. So that these the molecule have this whole protein structures is there. We have the different arms is there, and after dock we just have 2 different interactions there. And there is a one important thing in docking. We have to also visualize that, because most compound we have say that hydrophobicity and lipophilicity is very important. So hydrophobicity or lipophilicity, why it is important? Because they have to, I mean, because hydrophobicity is important to cross the biological membrane or cell membrane. And second thing is that hydrophobicity is also important for drug receptor interaction. So for this compound must have the partition coefficient log P high. And one more thing is that we can say also the lipophilic substituent constant, like chloro, fluoro, methyl. Like if you put the hydroxyl group or NH2 group, so log P is less. In that case, the drug will not able to cross the membrane. So this is all we have to calculate through the ADME analysis by quick prop in the Schrodinger. So we can see here, this is the XP visualization. There is a hydrophobic enclosure is there. So this is the whole molecule, and this area which are assigned as a blue stick, and here is the chloro is there. You can see this is the mounted towards this hydrophobic and surrounded by the hydrophobic amino acids is there. Similarly, here we can see this is the compound here, naphthalene area, the hydrophobic part. they have surrounded by the hydrophobic region. So during lead optimization, so if you putting like OH group, like NH2 group, so does it, they have the polar interaction, not non-polar interaction. In that case, DOC score is also going to decrease. And here we have also the naphthalene ring. Naphthalene is highly non-polar, hydrophobic is there. So in place of this, if you have to put certain group which are hydrophilic in nature, so these area may be present in the solvent area, exposed the solvent area, that the DOC score and activity is reduced is that. So hydrophobicity, and it is assumed that hydrophobicity is one of the important physiochemical properties which impart the lipophilicity of the molecule as well as the membrane permeability for their absorption in biological systems. So, so this compound, if you put this area, so here, this is the solvent exposed area. So during the design, if you have to remove this area by lead optimization, so might be your there is will be less likely to cause, I mean, polar interactions, and they have. So you have to remove this area. So that's why your DOC score, your bioactivity is just going to increase. So during the lead optimization, you have to remove this group, or you putting some important small group here, so that activity can be increased. And one more thing is that we also have to, by in XP visualization, we also see the rotatable bond is there, because due to rotatable bond in the compound form a different shape. So in that case, it is very difficult which pose of that compound is actually fit into the binding site. So that's why it's to avoid the much more rotation in that compound, we can provide this I mean, sterically rigid structures by incorporating certain double bond or certain groups, so that less likely to rotatable, and they have to form stable conformers. So this is all can we have to perform in the XP visualization. This is the review article we have published this last year in anti-cancer agent in medicinal chemistry. We have to perform the DPP4 inhibitor like adagliptin, lapigliptin. So they have the different binding pocket like S pocket, S2, and all that. So according to that, we have to design our compound. The binding energy is also important for that. This paper is published in anti-convergent of the GABA in 2016 or something, like in bioorganic chemistry. So we have to see the binding energy of different conformers of that ligand. So here you can see this is the top, which can in that binding area, some of the part is out of this, some of part is out of this. So this is the binding energy start from -20. So those compound which have the high binding energy in the negative, so they are more stable and more affinity for the binding site. We can also form the MMGBCA binding energy. So now we come to the molecular docking. So molecular dynamics simulation. So we have to understand the ligand binding ability in the active sites of the protein target. So once we have to perform the docking, what next we have to see how your molecule is actually stable within the biological system. Does it make a strong interaction with the receptor or not? So that can check by the RMSD. That can check by the, I mean, RMSD. Can you hear me? Hello? Hello? So here, I mean, so first in so in case of molecular dynamics, so first, can you hear me?
Yes, sir, you are audible.
Yes.
Hello.
Okay. Yes. Can you hear me now? I'm audible.
Yes, sir, you are audible.
Acha, thank you. Thank you. So first we have to see in molecular dynamics. So we have to check the stability of your compound at the binding site. So first thing in RMSD, we have to see that RMSD protein. This one is the RMSD of the protein on the left hand side, and the right hand side RMSD of the here is the ligand. So here is that orange color. This is called as the for protein, and the blue line which indicate that the your protein, and this one is the for ligand. Orange one is there. Okay. So now we have, how suppose if I'm going somewhere else through bus, and we have to hold some of my hands to the roof. So during the journey started from here New Delhi Jamia Hamdard to Old Delhi. So what we do during the jerking, during the movement, some of my hands grip is just lose some of that. So at the end of journey of 1 hour, lastly what we see, this hand is have 10% or 90% contact with that of the holdings. And the feet have the 10%, 30%. So we have to include only the stability of your ligand with the contact with that of the protein. Those who have less than 30% is just have to be avoid. So here in case of RMSD, so how much your ligand and proteins are moving, how much of your ligands and proteins are moving with the with respect to the standard or reference structure. So this is the co-crystal structures, and this one is the your design molecule. This paper has been I think going to be published within 1 or 2. This is under communications, and this is for the Jack kinase. So on March 2025 we have published one paper of the Jack kinase 3. You can Google it. So you can 2025 March we have published another paper of the Jack similar type of molecule. This is the under communication. Hopefully within 2 to 3 months is going to be published. So in x-axis, you can see the time which is in the nanosecond. Here in that x-second, nanosecond, which is start from 0 to 200 second, particularly time period for the run of these. And on the right hand side, ligand RMSD. So in y-axis you have seen that protein RMSD, and this one is the ligand RMSD. So in y-axis RMSD on the left side, so ligand RMSD shows as the here is the orange color, okay, and corresponds to that of the ligand RMSD. So blue, here is that blue color, which is the line corresponds to the protein RMSD. So what you want to see in the protein ligand RMSD. So ligand should not unbound. So this is the ligand should not unbound to the protein. So it should stay in the binding pocket. It should stay in the binding pocket. So if the RMSD of ligand is less than 2 or 3 angstroms, so here you can see this is from 1, and here this is the 2.5. So means if your RMSD is going beyond 3 angstroms, so that means your ligand is losing the grip from the protein. So that's why the ligand is not going to be stable, and they just have to not stay in the. So we cannot say stability of your ligand within the protein. So your RMSD must be less than 3 angstroms. Okay. So ligand RMSD less than, here is that of the 2.5. That's good, which shows that, which indicates that the ligand is staying in the binding pocket. But the protein RMSD is not beyond 3 angstroms, so that the protein is stable in the structure. So we can say the ligand and protein both are stable in the binding sites. So you got it. So now we come to the here that is called as the root mean square fluctuation. So here on the root mean square fluctuation is that before that I just missed one of these images, which is called as the secondary structure. Is that in that case we have to take the residue index, and here is called as the percentage secondary structure is there. So because they have the beta strand in the protein as well as the alpha helix in that protein, and they have the free region. I can show you later on. And next here is called as the root mean square. So how your protein is actually, how much which residue in the binding pocket of the amino acid is actually going to be more flexible. So beta strand or alpha helixes which are more flexible in their structure. So you can see in this area there's the more flexible. So flexibility, fluctuation should not be beyond, here 3 or something is that. On this is for the design molecule, this one, and this is for the co-crystal ligand with reference to co-crystal ligand. And after that, we also have to see this is the root mean square fluctuations of that particular protein. Similarly, I have just not incorporated one image that is called as the root mean square fluctuation of the ligand. So here, in case of ligand, suppose here this group is there, or something this group is there. So here, what in case of root mean square fluctuations of the ligand, so we have to assign these numbers in that particular atoms of your molecule, 1 2 3 4. Suppose here in this compound 30 atoms is there, so we have to stand from 1 to 30. Suppose here is the 30 carbon atom. So we see in case of root mean square fluctuation of ligand, 30 carbon number 30 is showing more fluctuation, which is beyond 3 angstrom of that. So in that case, yourself, you can have to change that particular group by different group, which then shows less fluctuation in the dynamic simulation. So this is also, dynamic is also the best way to develop the lead by changing certain group. And we can see, because our aim by the dynamics is that your molecule as well as the protein must be stable. Okay, so if some of that your assigned atom is showing more fluctuation in root mean square fluctuation of that ligand, so you can have to change through the optimizing that structures by incorporating. So, and after that you can further simulate these structures, revise structures or edited structure, and you see is there any change in the root mean square. Similarly, root mean square fluctuation of this is the protein. Similarly, we can perform the root mean square fluctuation of the ligand is there. So, and after that we have to see that call as the here you can see this is the amino acid, this is called as the here this is the hydrophobic amino acid, this is the water, and we just have to this is the negative charge, here is that positive charge, they have to make interaction. So here you can see that leucine of the co-crystal ligand, which shows the 93 or 85 make a hydrogen bond with that. Similarly here leucine 95, this is the hinge region of that map, JAK kinase 3. So you can see our design molecule as well as the co-crystal ligands have the hydrogen bond interaction, which has the 97%. During the simulation of 200 nanosecond, we see that 97% of your molecule showing stable stable interaction, hydrogen bonding. So which is here 93. So we can say, we can compare design molecule with respect to that of the reference molecule on the basis of dynamic simulation. Because if you perform the dock that here dock of this bis 220, so here the leucine 905 of the kinase of JAK kinase hinge region and all these amino acid, and this is the co-crystal ligand, all that leucine. So we can see that the dock binding pose is somehow same as that of the 2D leak plots of the after dynamic simulation, which shows that the same. Because in case of dynamic, we are not going to remove any of these water molecule, but in case of protein preparation wizard for docking, we have to delete the water beyond 5 angstrom. But if some of the water which has play a crucial role between the binding amino acids and the binding amino acid as well as the ligand, so which can retain those amino acid in the docking, but most of the time we have to delete or omit these water for effective interaction. So here you can see this is the reference compound. So these are the hydrophobic binding, and this is the hydrogen bonding. This one is the green color, blue color is that here, that this color is the hydrophobic interaction, this is for the ionic interaction is there. So similarly leucine 905 in the co-crystal as well as our design molecule is same as that. So this is also this paper is published in 2023. The same way we have the design molecules, and this one is the prototypic co-crystal drug. Here we also have the map kinase methionine 109, 93%. Here the 94% of the design molecule is there. So thank you all.
Hello, am I audible, sir?
Yes, thank you.
Yes. So thank you very much for this nice session on in silico approach in drug discovery. Sir, there are some questions from audience, so
Mhm.
I'm sharing that questions. Yes.
Mhm.
The first question is by Afrin Ansari. If there are multiple chains in macromolecules, we usually proceed with a single chain for docking. And how do we identify the chain involved in the binding site?
Yeah, Afreen, very good questions. Suppose we have to perform docking of any compound, we have to select the protein from the PDB-ID. And once we have to prepare the protein, it shows that A chain, B chain, C chain, or D chain. So, so visually you can inspect that where actually the co-crystal ligand is inbound in which chain, either the A chain, B chain, C chain, or D chain. So only you have to regain those chains for the protein preparation. Rest you can have to delete it. So if you are not going to delete the other chain, but you have to delete the cofactor or other the co-crystal or other ligand which are not useful for your docking purpose, you have to remove it. So you can perform the docking of by taking all the chain, regaining all the chain along with that of your co-crystal ligand. So automatically, once you have to perform the docking of your co-crystal, so automatically the system has remove that co-crystal, and the binding pocket becomes empty, and where you have to dock your design ligand molecule. So visually you can have to inspect by going to the protein data bank. Suppose I am taking here like 1M17 PDB ID. So here, this is the AQ4. The AQ4, this one is the structures of that, gefitinib or something like gefitinib. So this is in between the A chain or B chain. So either you have to, but this is AQ4. Sorry, this is AQ4 is the co-crystal which are present in that. So once you have to prepare the ligand, so you have to consider only those chain, not the C, D, A or other chain. So you have to either the mode to perform the dock. Okay, am I clear to Afrin?
Yes, sir. Yes.
Okay, thank you.
Sir, next question is by Professor Hemant Chikle. What parameters should we consider for homology modeling? What point should we have to add while communicating scientific paper?
So okay, because I would like not to go for the homology modeling, because in case of homology modeling, if you select some of your protein structures, okay, those which don't have any co-crystal inbound ligand is that, so in that case you have to take some of that bacterial species or other species which have their sequences there. So you by blast you have to match these sequences in the homology modeling, and you have to develop your own model, and you have to perform the doc. But what I suggest you, if you have the Solinger software, you go for the site map analysis, because in case of homology modeling, it take a lot of times from where you have to select those sequence and which species you have taken. It's a time taken, but homology modeling giving you more accurate as compared to the site map. Okay, thank you. I will share, you can give my email ID. I can share one of my papers to him where we have to perform the homology model. So I can share to that guy so he can understand by this way.
Yes, sir, definitely. Sir, another question by
What,
Dr. Umer.
what criteria is used to uh,
Yes. Another question
what
by
criteria is used to select a
yes, yes.
dock molecule in sitemap analysis? So if you in sitemap analysis already have explained how many site by taking one protein if your co-crystal ligand is not available. So these protein binding have a very uneven pocket is there. So you never know that which is the binding exact pocket of that. So in case of sitemap analysis, so once we have to generate a grid of particular site. So in sitemap you have to 1, 2, 3, 4 site is there. So already I've explained that there are site 1, site 2, site 3, site 4, and they have different values is there. One is the D score, another is the site score. Already I've explained how to calculate mathematically these. So D score means drugability score, which is more than one, we have to consider. So out of 5 site, some of these have the -9, -5, -4, sorry, 0.4, 0.8. Those D score, which is more than one, we have to consider those is there on the site score as well as the more. Here I'm just explaining here already. I mean,
Sir, one more question is there.
here you can see the gentleman. Here, this is the all sites of the EGFR. So this is the site map 1. Here the site score is more than 1. D score is more than 1. We rest of these we are not prepare any grid of these. This is the site 2, site 3, site 4, site 2. So these are not the exact binding site. These are the other allosteric site, because you know the binding site is always available in the deep pocket, and allosteric, yeah, other site which are present on the surface of that, because your molecule is hydrophobic. So more binding pocket is always in the deep, and they are hydrophobic in nature. But those which are present in the surface, they are the shallow structure. So these here also you can see in the surface. Here is also present in the surface. Here also it present on the surface. These are also the other site. They are the shallow structure site. So you cannot visually you can also see which is the binding site. You got it? So we have to consider only this for that. Those, if you have the D score is less than like 0.7 point, we have to discard it. How can I identify the active site or allosteric binding sites on the protein site? Because the allosteric binding site which are present on the surface, and and the allosteric, sorry, an active site or binding site, ATP binding site which are present in the deep. So I also explained recently we have to perform with the one of our Professor Javed Ali Pharmaceutical Department, which is under communications. You can see here those who are working in this. I mean, here this we have taken this uh, EGFR enzymes, and we have taken this PDBID 7JXM. So these are the all A, B, C, D chain is there. So here this is the, I mean, epigallocatechin, and this one is the salimerine is there. So this is on the deep pocket, this is on the surface is there. So you can see salimerine can binds with that of the, I mean, binding site of Egypt. So hydrogen bond interactions. So here you can see that this is the ATP binding site. This is the deep inside, and here is on the surface of that allosteric binding site. This is under communication. This work is, yes.
Okay, thank you very much, sir, for this informative session on in silico approaches in drug discovery and development. I am now here to summarizing your session. Sir, you are focusing in brief, in detail about protein ligand docking, mentioning various points with your case studies, including search algorithm, scoring function, then about structure-based and ligand-based drug design, then about induced feed docking and covalent ligand docking. Again, with your case studies, all of our delegates, no doubt, get knowledge about sitemap analysis, then molecular docking synergy, then computer-assisted drug synthesis, de novo design, then about presentation of docking data in various research papers, about virtual screening, about XP visualization, then about binding pockets by surface view models, energy, and very informative point of discussion is molecular dynamic simulations. So once again, I'm very much thankful to you, sir, for your informative session in clearing the concepts regarding in silico approaches in drug discovery and development. On the behalf of Internal Quality Assurance Cell of Gokhale Society, MS Gosavi, College of Pharmaceutical Education and Research, and APTI, I request you to accept this certificate of appreciation, sir.
Thank you, thank you. Actually, one thing I would like to thanks the audience who patiently listen my research work, because those research work which were published and few of them I have selected, and those which are undergoing, still I am in the learning phase. We cannot stop learning. So nowadays it's a era of AI, machine learning. So we just going to enter, and hopefully within 2 to 3 years, we might have to present on the AI based and machine learning talk if they just invite me in futures. Thank you so much.
Definitely,
Thank you,
sir.
Doctor.
Yes, sir. Sir, thank you very much for accepting our invitation and enlightening this session with your experience. Thank you once again, sir.
Thank you. And one thing I would like to share, during the COVID time, we just take my system to my home, and every day we, because my research area is synthesis of anti-cancer EGFR or anti-diabetic agent. Because this is the part time, you can say, because currently I am having 10 PhD scholar, 6 of them are getting fellowship, 2 of the project is undergoing. So my main aim is wet lab synthesis. So please don't only focus on that in silico. This is the side thing is there. If I well dressed, if I look good, so this is called as that hypothesis docking. This is not the core research. So core research is your wet lab. Still, if you are not learning the synthesis and all that, so this is very important. Thank you.
Thank you. Thank you very much, sir.